The headline on the other major news story today, to which we intend to devote some time, is very simple. Hello, Dolly.
It's possible we're seeing a scientific explosion comparable to the atom bomb or the moon rocket or DNA itself.
For many, it was a case of science gone too far.
Are we acting more like the creator than creatures? Are we trying to play the role of God in all this?
Predictions multiplied about just what this breakthrough would bring.
Soon it will be possible to give her thousands of absolutely identical sisters.
Animals could be cloned with human diseases and new therapies tested on them.
Whatever became of Dolly, and all that speculation about the brave new world she ushered in.
As science evolves, more and more radical discoveries are being made about the way that the world works. From transplanting human faces to doing complex surgeries with the use of just a few laser beams, the field of medicine has evolved very, very rapidly. And one of the most controversial topics that have risen with it is the topic of cloning. There are two main types of cloning. One is regenerative cloning, which is an offset of therapeutic cloning, and it's used to regenerate organs by cloning a few cells. And the second is reproductive cloning, which is cloning solely for the purpose of reproduction. Regenerative cloning has had a number of positive impacts in the field of medicine, specifically in transplant patients. On the other hand, reproductive cloning brings up a number of ethical implications that have to be considered when analyzing a topic as complex and controversial as cloning. A number of states have put up laws that prevent all cloning. Some laws prevent only some cloning, and some laws offer a lot of exceptions for specific cases. But in most cases, the laws are pretty ambiguous, and ambiguity is not the way to go when it comes to a topic as complex as cloning. Though the use of cloning can be advantageous in regenerative medicine, it can lead to a number of health problems when used in the reproductive sense, and it also raises a lot of ethical issues. I believe that a standard law should be enacted that bans reproductive cloning, but allows it in the field of regenerative science. In order to understand the whole cloning process, it's important to understand the basis of the science and how it works. So a short animation from McGraw-Hill that was found on Access Science, but that is narrated by me, can help us understand this process a little. The first step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with the restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its end that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme DNA ligase is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. July 5, 1996 was the date that Ian Wilmot, a Scottish scientist, received news that would change the world forever. Lamb number 6LL3, later named Dolly, was born, and Wilmot described his initial reaction as awe at the potential impact. With glass tubes about the thickness of a hair, over 400 attempts at this highly complicated procedure were conducted, but only one success was needed, and it was this one success that resulted in the birth of Dolly. The few people at Roslin who knew Dolly's secret were sworn to secrecy. They knew that if news of Dolly was leaked before it should be, it would spark an endless debate. Unfortunately for them, the news was leaked in the Sunday paper, and the news blew. Dolly's birth laid the groundwork for future cloning, and as you just saw, it raised a number of controversial issues. Before going into the actual debate, it's really important to understand the terminology associated with the next portion of the topic. Number one is cloning. Cloning is a form of reproduction in which offspring result not from chance but from the deliberate replication of the genetic makeup of another single individual. This is a form of asexual reproduction. The second is human cloning. Human cloning is the asexual production of a new human organism at all stages of development that is genetically virtually identical to a currently existing or previously existing human being. And the third is cloning to produce children. This is the production of a cloned human embryo that's formed solely for the purpose of initiating a pregnancy, with the ultimate goal of producing a child who will be genetically virtually identical to a currently existing or previously existing individual. A major concern in the cloning debate is the ethics of cloning. Two separate national-level reports depicted in a book entitled Cloning: Opposing Viewpoints concluded that attempts to clone a human being would be unethical, due to safety concerns and the likelihood of harm to those involved. Supporters of cloning say that cloning to produce children can serve several purposes. It would allow infertile couples or others to have genetically related children, allow the bearing of a child who could become an ideal transplant donor for a particular patient in need, enable a parent to keep a living connection with a dead spouse, or allow societies to try and replicate an individual with great talent. There are a number of issues with this. For starters, cloning to produce children would violate the principles of ethics of human research. Also, given the high rates of mortality in cloning other mammals, this process would be very unethical. Additionally, to find out a newer, less detrimental process, it would require a lot of experiments. In other words, there seems to be no ethical way to try to discover whether cloning to produce children can become safe. While human cloning, as is just discussed, is immoral, therapeutic cloning, I believe, is moral. Scientists in South Korea who created a new line of stem cells from cloned embryos have made a huge medical breakthrough in the possible treatment of many diseases. Researcher Alex Epstein says that this sort of technology is actually pro-life because it is still used to save human lives. He rejects that therapeutic cloning is immoral and stated that it would be very immoral not to use whatever technology is available, even therapeutic cloning, to improve life for human beings by opening doors to research that people didn't even know existed, and therefore discovering cures for currently incurable diseases. Therapeutic cloning is one of the most promising tools researchers have for developing therapies for treating diseases. Stem cells have the ability to transform into many other cells. This paves the way for more studies of diseases and future cures. Unfortunately, this process is strictly banned by a number of politicians because of their lack of understanding of the differences between reproductive and therapeutic cloning. Therapeutic cloning is a technique that transplants the nucleus containing the genetic material from an individual's own body cell, such as a skin cell, into an egg cell lacking a nucleus in order to produce totipotent stem cells. In principle, the product of these cells upon differentiation could become the nerve cell or heart cells that, upon transplantation, might replace a diseased tissue. This research offers hope to millions of Americans suffering from spinal cord injuries or diseases, such as heart failure, diabetes, Parkinson's, and Alzheimer's. In April of last year, a breakthrough in therapeutic cloning, using the human cloning technique, was used to create stem cells for adults for the first time. This could lead to tissues and organs being regrown, and the saving of many lives. Using the cloning technique that produced Dolly, researchers were able to turn an adult human skin cell into stem cells that can grow into any type of tissue in the body. They even managed to do this with a 75-year-old man, raising the prospect that body parts could be regenerated at an old age. This breakthrough could lead to new tissue transplant operations for a lot of debilitating disorders. Regardless, Dr. Lanza, co-author of the research, admitted that it was important for the future of regenerative medicine that research in therapeutic cloning should continue. Cloning, the word that had no meaning a few decades ago, can now mean a variety of things, from new organs to cloned armies. With science fiction movies and the media, people envisioned the future of cloning to result in millions of masked beings with guns, but fortunately, that's not the case. Cloning's impact in the field of medicine is vast, and therefore regenerative cloning should be legal. The future of cloning does not have to result in bad things. It can also result in cutting-edge procedures to find cures for diabetes, Alzheimer's, dementia, and cancer. Right laws and governmental support, there is no limit to the benefits that cloning can have on humanity.